Hello, my name is Michael Dabrowski, and with me I have
Stina Lundgren, Principal Project Advisor at Pelago.
And today we're going to talk about how you can navigate your chemistry and explore your biology with the CETSA method. And we're going to illustrate that with some case studies in both primary screening and target deconvolution. I'm going to kick off, and then I'm going to leave the word to Stina afterwards. So, Pelago is the home of CETSA. We're a specialized CRO focusing entirely on the CETSA method, and we also have the exclusive control of the IP of the method. And for continued development of the method and also its applications in drug discovery and diagnostics, we have a dedicated in-house R&D team to work on that. And we almost obsess about target engagement. As you know, that's the quantification of the relevant compound binding to the protein targets and at the intended site of action of the drug. And in order to develop a new drug, target engagement quantifications are crucial, obviously from the early stages of target ID and validation, but all the way through the development of your leads and into the clinic. If you don't know how much of the effect you can attribute to target engagement, it's difficult to take the right decisions to develop new and better drugs. And CETSA has indeed been used a lot in drug discovery, both for target-based applications, but also phenotypic applications to find new drugs. And the examples of applications range from the very early tool finding or tool compound profiling through mode of action, deconvolutions, selectivity, and translation, and even biomarker investigations. So it spans the whole range. The basis of the method is to treat, to heat, to separate, and then as a final step to detect protein. We have two base formats of the method, one that we call Navigate, or CETSA Navigate, which is antibody-based protein detection in the last step, and one that we call CETSA Explore, which is mass spectrometry-based detection at the last step. But common to both formats is that it works in any sample matrix that contains your biology and your proteins of interest. That can be both lysates and intact cells or even tissues, but also other species like bacteria or fungi or plants. First, we're going to focus on the Navigate format. And it takes advantage, or all CETSA formats takes advantage of the fact that if you heat shock a protein, the higher the temperature of the heat shock, the more protein you will denature. But if a drug or a ligand is bound to the protein, then this denaturation temperature or melting temperature will be different. And that's also used in the alpha CETSA assay, alpha CETSA HT assay, because the antibody coupled beads, they only bind to folded protein, which means that you will only have a signal when the two beads are proximal to each other. And obviously they won't be proximal to each other if they don't bind to such as denatured protein. So that's the way that the alpha CETSA assay separates between native protein and denatured protein. So the CETSA HT assay, we say that it's well suited for navigating your chemistry, and that's because you get, by using plates, the high throughputs. And you can use that on completely unmodified compounds, and also the protein that you're detecting is unmodified. So we use endogenously expressed protein, i.e. there is no cloning involved in this. And that combination of not modifying the proteins and selecting any sample matrix that contains your biology of interest, of course, gives you a very high physiological relevance of the output of your test or your assay. So in a bit more detail, how is this carried out? Well, again, you first treat, you now incubate your compounds with the cells in 96 to 384 well plates. You deliver the heat shock. break open the cells, and then you transfer the lysate to detection plate where you add the detection consumables, the antibody coupled beads. And then you can produce the melt curves, and a melt curve is first done without compound and then again with compound. And if you see a change in amount of protein left after the heat shock, this will look like a shift in the curve. And here you can see clear shifts between the melting curves. If you see such a shift, that means that the compound is engaging with your target. And obviously is detected, and therefore you can go ahead and start screening for EC50 temperature, EC50 values, but you do that at a single temperature. So here on the left-hand side, you see example of using such a single screening temperature, varying the concentration of the drug, and thereby obtaining concentration response curves, which is the target engagement potency of your compound on that protein. And two of our clients have done exactly that. We developed the assay, and then Vertex screened in the first case here a relatively small library, but they did that because they were really interested in this challenging multi-protein target, multi-protein complex target, and they had no tools to prosecute this target. So after developing the assay, they then screened this relatively small library, and indeed this assay developed or delivered new chemical scaffolds which weren't identified in other screens, and therefore Vertex can now prosecute this interesting target. AstraZeneca did one step further, at least in terms of the numbers of compound screens, because they screened 500,000 compounds against CRAF. And of course, the assay identified both the known CRAF chemistry, but also identified new chemical starting points for this very well-validated oncology target. And as an added bonus, the CETSA assay in this case also showed a very low liability towards pains. So that's what customers done. I'll now bring Stina back in, and she'll talk to you about applications of the method.
Thank you, Michael. So I'm going to start off by telling you about the project we have done internally at Pelago. So we have conducted a primary screen on CDK4 and STING. We started this project by validating these assays, and we chose THP1 cells and K562 cells as appropriate cells to do the assay in. We screened some different temperature to select a good temperature for the screening, and we used reference compound for this. For the compound library, we selected a compound library of 11,000 compounds. It was the same library that was screened against both targets. This was a focus library, and it included lead-like and drug-like compounds. We also included the Prestwick library, and also part of the library were fragments. We did a pre-screening where we checked the robustness of the assay and did Z prime calculations at the different temperatures. And in the end, we did a compound screening of 37 assay plates. These were pre-spotted, and we used a concentration of 50 micromolar for the screening. Here you see the data that came out of the pre-screen. So for CDK4, we selected THP1 cells for the screen, and 46 degrees was selected as the screening temperature. We used a number of known CDK4 inhibitors as reference compound. And the 46 degrees temperature was selected on the temperature where we could see a good shift of the melt curve with this reference compound. The assay was really robust, and we had a Z prime factor of 0.9 for this assay. For for STING, we selected K562 as the cell matrix to use, and we included both agonist and antagonist as reference compounds. Interestingly, the agonists were able to stabilize STING, whereas the two STING inhibitors, so the two antagonists, were destabilizing STING. 51 degrees was selected as the screening temperature, and this was selected because we thought we would be able to both pick up protein hits that were able to stabilize STING at this temperature at the same time as capturing hits that would destabilize the protein. This was also a robust assay, and Z prime was of 0.9 for the pre-screen. Moving into the primary screening, so this is the results from the screen. And as you can see, there are a fair amount of compounds that are able to stabilize STING. But when we look at the compounds that are destabilizing, there are really many hits. So it seems like STING is fairly sensitive, and something in this assay makes it prone to be destabilized by a bound compound treatment. That is not true for CDK4. In this, we had very few compounds that were able to destabilize CDK4, whereas the majority of hits came from stabilizing mode of action. So looking at the compound selection flowchart, we did a primary screen of 11,000 compound at 50 macromolar. The temperature was 46 for CDK4 and 51 for STING. CDK4 had 137 compounds that were stabilizing the protein and 39 that was destabilizing the CDK4. So these were viewed as primary hits, preliminary hits. For STING, we had 197 compounds that were stabilizing the protein, and almost 2,000 compounds were destabilizing and viewed as preliminary hits. For the next step, we set up a confirmation assay. And in this, we decided to screen at 3 different concentrations. So 50, 10, and 2.5 macromolar, and then using the same screening temperatures as in the primary assay. Around 2,200 compounds were screened, and the same compounds were screened against both targets. This is data from the CDK4 hit confirmation assay. And in this plot, we are correlating the primary screening with this data that came out of the confirmation assay. So these are both from the 50 macromolar compound screening. And as you can see, there's a good correlation between the 2 rounds, and the assay are showing good reproducibility. So looking at the hit rate for CDK4, we had the hit rate in the primary assay for the stabilizing compounds around 1.2 and 0.4 for the destabilizing compound hits. And when we have reviewed the data from the confirmation assay, taking into consider all 3 concentration points, around 80 to 90% of the preliminary hits have been confirmed. The next step, we're planning to do concentration response curves, so 10 different concentrations, and also potentially following this up with some functional assays. And this is also in line how CETSA have been used by others, and that's CETSA HD for hit confirmation. So first of all, there is this biochemical FB assay and the correlation with the CETSA data. And in this graph, some compounds were not able to engage the target in the cellular environment. This could be either due to low permeability or for some other reason not engaging the target in intact cells. Secondly, there's a correlation between the CETSA HD data and a functional assay, in this case, cellular phosphorylation assay. And first of all, I could say there's a good correlation. And of course, this is something that strengthens the target validation. So giving you confidence that the effect you see in the functional assay is due to compounds acting on the target. There was a number of silent binders that are identified in the CETSA HD assay. And this, of course, not so interesting if you're targeting the cellular effect. but they could be interesting starting point if your project is, for example, a PROTACs project, where you want to identify binders that you can link into an E3 ligand binding motif. Secondly, I will move into talk a little bit more about the case study we did last year at Pelago on MEC1. So this is an alpha CETSA HD assay where we have profiled a number of MEC1 reference compounds. We included both compounds that were binding to the ATP competitive. They were ATP competitive, so they were binding to the ATP pocket, but also allosteric binders. They were non-ATP competitive. And the allosteric compounds were all able to stabilize MEC1 upon compound treatment, whereas the ATP competitive compound were giving a destabilization effect on MEC1. Next, we wanted to study if we could measure the inhibitory activity and target engagement in parallel. So looking at MEC1 as part of the RAS-RAF pathway, MEC1 is phosphorylating ERK. We did a study in A549 cells, and the first data we generated was the CETSA EC50, so a dose response experiment at a single temperature, in this case 54 degrees. The data you see here is with celiptenib, one of the allosteric compounds. Secondly, we looked at the level of phosphorylated ERK, where we could see that the amount of phosphorylated ERK were decreased by increased compound concentration. And then following as a control experiment, we also looked at the total level of ERK, which were non-affected with the increased compound concentration. We did the same study with the ATP competitive compound. That was, as I said before, destabilizing MEC1 upon compound treatment. But in the same manner as the allosteric compound, we're also affecting the level of phosphorylated ERK, but not affecting the total level of ERK. We also profiled PROTACs using the CETSA HD format. So as you might all be aware, the PROTAC is consisting of a ligand binding to the E3 ligase and then connected with a linker to a warhead. And this is then bringing the target protein in close proximity to the E3 ligase, which is ubiquitinating the target and marking for degradation by the proteasome. So in this study, we looked at some CDK4 PROTACs. They are then based on ligand binding to cereblon, a linker, and then a warhead. And then these two PROTACs are one based on palbociclib and the second one on ribociclib. And looking at the melt and shift curves, we could confirm that both of these PROTACs were able to engage CDK4. And we also included the reference compound palbociclib, which also were able to stabilize CDK4 upon compound treatment. We followed this up also by doing a dose response at 46 degrees and could confirm the same event. Secondly, we move into looking at target engagement towards the E3 ligase, so in this case, cereblon. And in this study, we also included a third PROTAC, a BRD4 PROTAC, which has, but also using the same E3 ligase, cereblon, for the degradation. And in this study, we could also confirm that all these three PROTACs were able to bind to cereblon and engage it in intact cells. Now I'm going to change subject and move away from the CETSA Navigate, the targeted format of CETSA, and focus on the CETSA Explore. So the mass spec-based CETSA format, which is a protein-wide format that is suitable for target deconvolution, as well as mode of action studies and selectivity determinations. So the assay protocol for CETSA-MS starts with incubating cells or lysate or even tissue, with and without the compound. Then applying heat shock, followed by centrifugation. After the centrifugation, the soluble fractions are analyzed on a mass spectrometer. And from this data, we can generate thermal shift data, and we can then identify proteins that have either been stabilized or destabilized at protein heads. And this can then be visualized in a volcano plot and looking at the protein profiling. So in a single CETSA-MS experiment, we typically report on around 7,000 proteins. A majority of these proteins are typically not affected in their thermal stability. This means there is neither been a stabilization nor a destabilization event happening. But then we have a group of proteins that have been affected. And that could be either proteins that are affected in the thermal stability by direct binding of the compounds, and this could be, of course, both primary as well as off targets. In addition to this, we also have in the intact cell experiment, proteins that have been affected downstreams. So this is associated proteins that are being affected in the thermal stability. And this can be due to loss of interaction partners, phosphorylation events, dimerizations. And this group is not present in the lysate experiment. So this can look like this. This is an example with pomalidomide in K562 cells. First is the lysate data, where the biology is off. So in this, we see direct binding proteins being affected. And then comparing this to the intact cell, we have more hits. So in addition to cereblon, we see a number of other proteins being affected. And these are not direct binding proteins, but part of the pathway. Another example of this is this case, where we have profiled staurosporin. This is a pan-kinase inhibitor, and there's no surprise there are many proteins being affected in the lysate. A majority of these proteins are kinases. And then adding to this, the data in intact cells, where there are even more proteins being affected. A majority of the additional proteins in the intact data are actually proteins that are downstream of the kinases. So proteins that are, for example, phosphorylated by kinase and thereby being affected in thermal stability when we are applying staurosporin. Another way of illustrating this data is to look at the kinase family tree. So in this plot, the blue round circles are the proteins that are stabilized by staurosporin. And then we have the pink ones that are being destabilized by staurosporin. And in addition to this, we have also correlated the reported KD values from a competitive binding assay with the CETSA EC50 we have been able to generate in the CETSA-MS experiment. And as you can see, there's a really good correlation between these two. And this highlights the value of using CETSA-MS for assessing the selectivity of your compounds. Now moving to a more selective kinase inhibitor, or coming back to palbociclib. So this is the lysate data and the intact cell data. Starting with the lysate data, the proteins targeted are being affected in the thermal stability is mainly kinase off targets of this CDK4, CDK6 inhibitor. Then comparing that with intact data, we see in addition to the kinase proteins being affected, also some of the proteins in the pathway. One example of this is RB1, which is known to be phosphorylated by CDKs. And this event is then inhibited by palbociclib. We have also profiled the PROTACs in the CETSA-MS format. So this is the same PROTAC we included in the CETSA HT study I described earlier. The first data here is the lysate data. So after 15 minutes incubation at room temperature, looking at the CETSA MS profiling in K562 cells. Both CDK6 and 4 are among the protein hits, in addition to a number of kinase targets that can be viewed as the off targets of palbociclib. Secondly, you see the CETSA MS data in intact K562 cells, and this is after 1 hour compound incubation. And in this data set, we no longer see CDK4 nor 6 as protein targets. The E3 ligase cereblon are present in both data sets, so both in the lysate and the intact, but the 2 primary targets are not included in the intact data set. We followed this up by doing a quantitative proteomics in K562 cells, looking at the data after 1 and 6 hours at 36 degrees. And we could clearly see that we had an effect on the abundance level of these 2 primary targets. And there were not any surprise that these 2 were being degraded upon the PROTACs acting as it should. We also identified 1 protein that had an increased abundance. This was a cage-like protein 8, which was had an increased abundance upon compound treatment. So coming back to the wheel Michael showed earlier and concluding this webinar, CETSA HT can be used for primary screening, but also for tool compound generation, hit confirmation, SAR generation, but also in translational studies. It's a sensitive and robust assay, and it allows both for high and low affinity binders to be identified. We also shown that it can be used for profiling the target engagement of PROTACs, and that is by both assessing the target engagement of the target protein as well as the E3 ligase. The CETSA MS format can be used for studying both direct binding as well as pathway included in the downstream cellular pathways effect. And the application range from selectivity and specificity studies, as well as assessing the mode of action and target deconvolution. And you can also use it for tool and probe compound validation. So I hope you enjoyed this webinar and want to learn more. I encourage you to visit our booth and download some of our app notes and article, as well as a list of targets that we have previously worked on. You can also book a one-to-one meeting with us, and for that, I encourage you to reach out to my colleague, Ylva Elias, she's a business development manager, and we'll be happy to schedule a meeting with you. And finally, I would like to thank you for your kind attention, and ending this with a skiing photo. So while we're not engaging ourselves in running target engagement assay in the lab, we're engaging ourselves in winter activities. Thank you.
Thank you for your attention.
These people are scientists, directors, and portfolio managers. This guy is a drug discovery project leader. They have one thing in common. They all failed to check relevant target engagement early in their projects, so they can't see where they're heading. That's risky. Ouch. Let's make sure it doesn't happen in your project.